Okay, so I'm going to introduce Dr. Greg Schultz. Dr. Greg Schultz is a professor emeritus at the University of Florida, where he serves as the director of the Institute for Wound Research and professor of obstetrics and gynecology. Dr. Greg Schultz was flying back from an event and just flew overnight, so he will be joining us for the roundtable. However, I will be loading up his presentation, which he has recorded, and then you can ask any questions that you might have of him in the roundtable. I want to thank everybody for being here today, and I want to again give a shout out to NATROX for their generous unrestricted educational grant. We'll give you more information on certificates and how to obtain your CME certificate after the event. So I wanted to take a minute to tell you that as well. So I'm going to load up Dr. Schultz's presentation and enjoy, and I'll see you after.
Hello, everyone. I'm Gregory Schultz, Professor Emeritus at the University of Florida, where I was the director of the Institute for Wound Research. And I'm currently the Chief Science Officer for Inotech and the NATROX product. My goal is to describe the science behind the clinical impact of oxygen in wounds. It's probably appropriate to start by just emphasizing that oxygen plays critical roles in all phases of wound healing. And so, for example, we're going to look at the role of oxygen in both angiogenesis and revascularization, especially as you can appreciate cell metabolism and cell proliferation and energy production. We'll look at the critical role that oxygen plays in producing antibacterial activities in neutrophils and macrophages. We'll also examine the critical role that oxygen plays in both collagen synthesis and especially in stabilizing and strengthening the new collagen molecules that are made in the wound bed. And obviously also I'll show you data that topical oxygen therapy can correct many of the problems of both ischemia as well as diabetes in animal models, and will stimulate cell proliferation and reepithelialization. It's probably appropriate to start by just looking at the partial pressure of oxygen in both chronic and acute wounds, because it can vary tremendously. And if we look at typical oxygen tensions as revealed and indicated by the near-infrared spectroscopy that can detect and quantitate the level of oxygenated hemoglobin as well as oxygen in other components within the wound. But basically, the tissue partial pressure of oxygen, which you'll often see abbreviated as PO2, will vary between different wounds and even within the same wound. And in normal tissue, the oxygen tension is usually around 100 milligrams of mercury, but in the center of wounds, it can even be as low as 10 milligrams of mercury. And using the fluorescence near-infrared spectroscopy that you see in the right-hand panels. If we look at just normal visible light before continuous topical oxygen therapy, you can see the wound bed only has about 58% saturation with oxygenated hemoglobin. After 3 weeks of topical oxygen therapy, you can see that it's increased significantly up to about 78%, which shows the ability of continuous topical oxygen therapy to make a very significant physiological impact on the wound bed and wound cells. Another critical factor that we want to look at is the critical role of oxygen in infection. And so, for example, as you're very much aware, in our normal immune cells, our neutrophils and macrophages, when they encounter a bacteria, they will invaginate it in a phagosome, and this activates an NADPH enzyme complex in the membrane that generates reactive oxygen molecules, especially initially as the oxygen radical O2 dot, which will lead eventually to production of both hydrogen peroxide and hypochlorous acid. This generation of the microbicidal reactive oxygen species is called the oxidative burst. As I said, will generate both hydrogen peroxide and hypochlorous acid. In addition, it will generate nitric oxide, NO, which is a reactive nitrogen species that has both anti-biofilm activity, but also has very important effects on vasodilation and angiogenesis. So if we just do a little bit of quantitation on the production of the superoxide radical, the O2 dot, which is the free electron, and we can see that it very substantially depends on both the partial oxygen pressure and the pH. So if we do laboratory experiments where we take activated neutrophils in culture and we look at the consumption of oxygen with different concentrations of oxygen in the medium, you can see that the Km, about the half saturation of this occurs around about 80 millimeters of mercury. Down here around 10 to 15 millimeters, which we see in many of the ischemic wounds, there's very little oxygen consumption. That's because there's very little superoxide generation by the activated neutrophils at this low oxygen, partial pressure of oxygen around 15, because again, we want to be up here somewhere around 100 millimeters. And this eventually generates the true superoxide radical that generates the reactive oxygen species. And again, this is very dependent on pH. So again, the pH around 7 gives us a nearly optimal production of the superoxide radical. So we see that the ability of generation of bactericidal capacity will raise 3 to 4-fold if the partial pressure of oxygen in a wound is raised from about 15 millimeters of mercury up to around 100 or higher, and especially if the pH is in a more normal range. Another important aspect for us to realize is that when the percentage of bacteria in venous diabetic or pressure ulcer samples is analyzed and determined by quantitative PCR analysis, as reported initially by Scott Dowd and Randy Walcott, you can see that in pressure ulcers, over 60% of the bacterial population are strict anaerobes, which means that this allows, their environments allow these strict anaerobes to grow only when the oxygen concentration is at a very low level. So obviously we want to get rid of these anaerobes, and one way to do that is by using continuous topical oxygen therapy. So in a initial chronic diabetic foot ulcer studies using continuous topical oxygen therapy, the authors found that there was a reduction in the anaerobic bacteria and a shift to more aerobes and facultative anaerobes observed over the 8 weeks of treatment in the wounds that were healing. And this led to a greater diversity of the microbiome, again, reported in the ulcers that healed, whereas there was no significant diversity change when the wounds were non-healing. And this shows that we want to be able to shift the proportion of bacteria and types of bacteria when the wounds are not healing, using topical oxygen to shift it into a proportion and a distribution of microbiome that's more conducive with healing. Now, one of the biggest issues that has emerged in the last 10 years or so that helps to understand the critical role that the microbiome plays in impairing healing is the presence of biofilms, which have been identified in a meta-analysis by Matt Malone that there's about 80% of the biopsies in chronic wounds typically have structures as shown in the scanning EM of a biopsy at the surface of a chronic wound, but only a very low percentage of acute wounds that are healing actually contain these biofilm type structures. And one of the key things that we'll look at is that biofilms are able to help evade our immune cells, neutrophils and macrophages, as well as, importantly, that metabolically dormant bacteria and biofilms are tolerant to most antibiotics and antimicrobials, as I'll show you the reasons for that. So again, why are biofilms tolerant to many of the antimicrobials that are used both systemically and topically? Well, one of the key factors, and there are multiple, but one of the key factors is that bacteria and biofilms grow very slowly. That is, they have very slow metabolism. And one reason that happens is that the oxygen concentration decreases with depth as you go into a biofilm. So data from Dr. Studley's group showed that when they measured oxygen concentrations in laboratory models of biofilms, that there's obviously abundant oxygen at the surface, but as you go down even about 50 to 60 microns of depth of the mature biofilm, there really becomes an incredibly ischemic area of low oxygen concentration. This is where a lot of those strict anaerobic bacteria find a niche to grow. And again, absolutely critical to understand is that the reduced metabolic activity of these bugs that are in an oxygen deficient area have low metabolic activity, and antibiotics only effectively kill metabolically active bacteria, such that the reduced metabolism causes this reduced efficiency in killing by the antibiotics. So really killing, especially of aerobic and facultative anaerobics, are oxygen dependent. And this happens because the antibiotics are able, normally they target and inhibit specific bacterial enzyme systems, such as influx and efflux into the bacteria, and they will, the antibiotics will do such things as block the synthesis of proteins or the synthesis of DNA or lipids, and this is what actually then leads to killing of the metabolically active bacteria. So biofilms and antimicrobials, is there a role for oxygen? What the data from laboratory and animal models are beginning to strongly show is that supplemental oxygen has been shown to increase oxygen penetration into a biofilm. So that provides oxygen in those ischemic areas of the thick biofilm matrix. And this additional oxygen, as I'll show you, actually wakes up metabolically dormant biofilm bacteria, and they start to grow, which means they're increasing metabolism. And that means that the biofilm bacteria become more susceptible to the antibiotics because the antibiotics work on metabolically active bacteria. This leads to the interpretation that adjunctive oxygen may increase the effects of antibiotics and other antimicrobials against biofilms in chronic wounds. And this can help to manage infections more effectively and to use antimicrobials more appropriately in treating these chronic wounds. I've told you that the topical oxygen will increase the killing of mature bacteria in biofilms. Let me show you a little bit of data. This is using mature biofilms grown in the porcine dermal explant model, where the bacteria are grown actually on the dermis of pigskin explants. This is the best laboratory-based ex vivo model we have for biofilm growth in chronic wounds. And what you can see in terms of the concentration of the bacteria that are recovered, no treatment, there's a very high level, 10 to the 9th CFUs per biopsy. If we use just the oxygen for Pseudomonas, there's a small reduction. If we use only Neosporin on these, there's again only about 1 log. But if we combine oxygen with the antibiotic, the Neosporin, in the Pseudomonas and especially in the Staph aureus, we'll get 3 to 5 logs additional killing compared to the antibiotic by itself. So this shows a stimulation of the bacteria in the biofilm to make them more sensitive to the killing of a very common broad-acting antibiotic. Well, why does that happen? Well, not to get too deep into the molecular biology, but basically using these dermal explants that are inoculated with Pseudomonas, then exposed for continuous topical oxygen therapy for 24 hours, and measuring the expression of 7 Pseudomonas aeruginosa genes that are involved in biofilm formation and quorum protein secretion, as well as general metabolism, using the reverse transcription quantitative PCR, which is state of the art. What is shown graphically here is that the continuous topical oxygen therapy upregulated transcription of these 3 genes that are involved in metabolic activity within the bacterial cells in the biofilm following the continuous topical oxygen treatment. Also importantly, the CTOC downregulated transcription of these 3 genes that are key involved in biofilm formation. The interpretation of these data from the lab is that the continuous topical oxygen therapy may help increase the sensitivity of bacteria and biofilms to antibiotics by stimulating the metabolic activity of dormant bacteria and helping to disperse mature biofilm communities. Another key aspect of the general actions of oxygen is in the production of energy, of ATP. And you're all familiar, remember back to biochemistry when glucose is taken up by cells and undergoes just the normal glycolysis initial steps producing pyruvate. If there's no oxygen, basically that's where the energy production stops and you have lactic acid and ethanol production. That only generates 2 ATPs per glucose molecule. In contrast, if there is adequate oxygen, then the pyruvate gets entered into the Krebs cycle, and with the oxygen, there is a huge increase in ATP production. In other words, 34 additional ATP molecules can be generated from the original single glucose molecule, giving 36 total ATPs produced in the mitochondria when there is adequate oxygen compared to just 2 when there is not adequate oxygen. So this shows the tremendous increase in energy production that all of the cells are going to have to need and use in normal wound healing. Well, I've shown you data. What about some animal models? Well, if we use the rabbit ear ischemic wound model, Tom Musto's group reported that if the wound epithelialization, the coverage of these full thickness injuries in the ischemic rabbit ear model measured at day 5 compared to just normal controls with occlusive dressings, that there's a 91%, almost a doubling of the amount of epithelial coverage at day 5, and by day 8 there's a 156% increase. And these histological analysis showed then that there was greater epithelial coverage in the oxygen treatment versus control at both day 5 and day 8. Okay, that was an ischemic model. What about a diabetic model? So using the mouse model of diabetic skin wounds, again, very similar positive benefits shown in terms of epithelialization compared to just occlusive control. So on day 6, as shown in these images of control or oxygen treated, there's a 60% epithelialization versus 45% in the control. At day 10, there's 83% in topical oxygen treatment versus 71% in the control. And importantly, complete reepithelialization at day 10 was in 57% of the continuous topical oxygen treated wounds versus only 25% in control. Now, another critical aspect that we'll look at a little bit more detail in the following slide is that collagen remodeling, that is the formation of the scar and then the remodeling of the scar, is extremely dependent optimally on oxygen levels. So there's a significant increase histologically with deposition of collagen at the base of the wound after topical oxygen therapy, and they appear to be more organized and fibrillous in appearance, suggesting that the remodeling is significantly more advanced than controls when adequate oxygen is present. Well, one of the ways that happens is, you remember back in protein synthesis, we have messenger RNA that gets transcribed on the ribosomes and makes the collagen peptide chains, and 3 of those collagen peptide chains come together. They form the initial pro-collagen molecule that assembles, and then the ends, the pro-collagen molecule ends get cut off, and they assemble into these staggered collagen fibrils made up of multiple collagen fibers. But what's important for us to realize is that for these initial collagen fibrils to be stabilized, they have to have covalent bonds formed between the lysine and proline groups at the end of the collagen fiber molecules. So collagen deposition, polymerization, these enzymes, the prolyl hydroxylase and lysyl hydroxylase, are required for cross-linking, and that requires optimal levels of oxygen because these hydroxylase enzymes require oxygen as a cofactor. So in low oxygen, the collagen hydroxylation and maturation is reduced and collagen remains fragile. That is, it is not highly stable. And this really impairs new vessels because they tend to break and disintegrate. In contrast, when collagen deposition proceeds with adequate oxygen, the new vessels are stable and they are produced at the highest possible growth when there is adequate oxygen in the tissue. So it's kind of a summary. Collagen formed in a hypoxic environment is only about 30% as strong as collagen formed in normal oxygen concentrations. So in summary, what I hope I've done is to show you the critical roles that oxygen plays in multiple essential roles and activities in wound healing. So, and also, unfortunately, oxygen levels vary greatly in acute and especially across chronic wound tissues. And continuous topical oxygen therapy markedly increases the partial pressure oxygen levels in wound tissue. What that does is the oxygen supports the immune response by generating these reactive oxygen species, and that dramatically enhances the ability to fight infections, as I showed you. It increases the bacterial biofilm metabolism, which increases the efficiency of antibiotics in killing the now activated biofilm bacteria. It produces tremendously more, 18 times more energy than in ischemic wounds, which allow the cells to function properly. That results in increased cell signaling and migration, resulting in revascularization, as I showed you, reepithelialization, and that accelerates and improves wound healing, and especially when we look at collagen maturation and remodeling, because this enhances the tissue strength and durability of the scar tissue in the new wound, which leads to less recurrence. So what I hope I've done is show you some of the critical roles that oxygen plays and the ability to increase oxygen effective concentrations in wounds using the continuous topical oxygen therapy. I look forward to answering questions.
Well done.
Hello, everyone. I'm Gregory Schultz,
Got it.
Professor Emeritus at the University of Florida, where
Okay, we don't need that one again. Sorry. Excellent presentation by Dr. Schultz. He will be in the roundtable live to answer any questions that you might have. I want to thank once again NATROX for their unrestricted educational grant. I think we've learned a lot about continuous topical oxygen therapy, the use, the mechanism, patient compliance, and a lot of other things from the previous speakers. They'll all be in the roundtable as well. There is an exhibit hall that you can go check out the NATROX booth. If you'd like, you just go back to the lobby, click on exhibit hall, and you can download some videos, documents, papers, things of that nature, watch them later. You can also log into this event and review any of the content that you have seen today. We don't take it down. And you can refer any of your friends, clinicians, anybody that you think could benefit from this content. They can also join and watch the recordings as well. Regarding CME credits, will be after the event, after we tally up all of the attendance records, we'll submit a link for you to click and you can download your certificate. So that'll be really easy for you to do. I think you'll like that process. Definitely like that process. So I'm going to close this session out. The roundtable is beginning in about less than a minute. So we're going to pop over to that room and I'll see you all there. And once again, thank you all for being here. It's been an awesome event thus far. So looking to close it up with the roundtable and answer all of your questions and then some new ones as well. Thanks a lot, and we'll see you in a little bit.